Sorry, it's not opening.
Hmm?
My net is so slow.
So be responsive on like the mic, unmute yourself and speak. The rest of the students, switch on your videos quickly. Why is no one listening to me? I don't understand. Marva, switch on your video. So in the previous class, we were discussing about the lac operon, the whole entire concept about the operon, right? So today we will discuss more on how actually the lac operon basically functions. So lac operon, it basically is how the metabolism of the lactose would take place in the case of the bacteria. Okay, so when we talk about the lac operon, we have seen that there are 3 structural genes present in them. And accordingly, there are different genes that synthesize the different products that are required by the bacteria, or you can say the enzymes that the particular gene will be coding. So the Z gene, it codes for the enzyme beta galactosidase, and this enzyme, it is responsible for breaking down the lactose into its components, that is galactose and glucose. The LAC-Y is the one that is coding for the permease enzyme, and this one will increase the permeability. Like it's there in the name only, permease, permeability. So it increases the permeability of the cell to beta galactosides. And lastly, the LAC-A, it synthesizes the enzyme, or it codes for the enzyme trans-acetylase, which will be transferring the acetyl group to the beta galactoside. So these are the 3 structural genes that are present in the case of the eukaryote, the bacteria, the prokaryote. Now here the operator gene, what will it do? It will interact with the protein or the regulator molecule, meaning either it will interact with it and stop the process or continue the process. Then there is a promoter gene, which is the part where the RNA polymerase attaches, and then the regulatory gene, this gene. Do you see this one? So this I gene is the one that will either, it will code for the repressor protein, okay? And if, let's just say, in this medium, when lactose is absent, okay? So in that case, what happens? This repressor gene will produce the repressor protein, or you can say it will produce the repressor RNA, which will form the repressor molecule, which is a protein. Now, as soon as this repressor molecule will be formed, it will go and it will sit over here and it will bind with the operator region. Now, when the operator region has this particular repressor binded into it, then the repressor, like the RNA polymerase, will not be able to transcribe the 3 structural genes, and then those enzymes will not be formed. And this happens in the case of when the inducer, that is the lactose, is absent. Clear? When it is absent, the repressor protein will be synthesized and the whole entire process will be inactivated. Okay? So if you see here, initially, when the repressor protein and the operator gene combines, the whole entire mechanism gets switched off. So this is the negative control or regulation. Okay, because in this case, when the inducer is absent, then what will happen? This first situation is going to take place. Now, the second scenario is the presence of inducer. Now, what happens is that now the lactose is present. So this regulator one, it is going to, or this repressor gene is going to synthesize the repressor mRNA, which will make the repressor protein normal. But now, since there is the presence of inducer, this inducer will go and bind with the repressor. So now the repressor protein has a change in the shape, as a result of which it is not able to bind at the operator region. And since it is not able to bind, then the RNA polymerase and comes bind, and all of these things, they get first transcription of it happens, then translation happens, and ultimately the galactosidase, the permease, and all of these three enzymes are formed. So always remember here inducer refers to the lactose. If the lactose is provided in the medium, then the process will take place. This is the second scenario. And if the lactose is absent, or so in that case, the repressor gene is going to bind with the operator region. Is it clear, students? Did you people get it? How it's actually happening and why is it called as the negative control? Why is it called as the negative control? Can someone explain? Why do you think it is called as the negative control? See, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of the permease. A very low level of expression of lac operon has to be present in such cell at that time because lactose cannot enter those cells. Now, when the inducer is present, such as if the lactose is present in the medium, or you add allolactose, which is like just like a form of lactose, then what happens? The repressor is getting inactivated by the interaction with the inducer. Inducer means the lactose or the allolactose, as a result of which the RNA polymerase binds to the promoter and the transcription happens. Now tell me one thing, students, here this part. They are saying first this Z, Y and A, it undergoes the process of transcription and then it undergoes the process of translation. Can someone just explain what is happening over here? Who can explain it to me? Anyone, whoever knows how to explain it, you can go for it. Yes, why is no one responding? Can you people hear me? Aisha, your video is stuck. Jobita, am I audible?
Yes, ma'am.
Okay. So what happened? Can someone explain this to me? Muniba, can you explain it to me?
Because the product of transcription is the messenger RNA,
Hmm, that's
so
true.
it
Very good.
further starts the materials for translation, the process
Huh,
of translation.
yes. What, like in layman term, what do you call the process of translation as?
And sorry, can you repeat?
Hmm?
Can you repeat?
I'm saying in layman terms, what is another name of translation? Like normal name of translation? Translation means
Protein
protein.
synthesis.
Yes. Means protein synthesis. So whenever the proteins are formed, then that is called as translation. So basically during this, what happens is that the whole entire thing is like ultimately these enzymes, they are supposed to be the proteins only. So that is why in order for them to be formed, the translation happens and translation happens on the RNA. That is why this whole entire thing is happening. This is your lac operon. Does anyone have any doubts up until now?
No,
Ma'am, can
ma'am.
you repeat?
Yes, Marwa, any doubts?
Ma'am, can you just once again repeat?
Which one? The whole entire thing?
No, ma'am, the one now you talk about.
Oh, right now. See, right now I said that these structural genes, ultimately they have to synthesize a protein. Now see, there is no gene that will directly make the protein. First of all, that gene will undergo the process of transcription and make the necessary mRNA. And when that mRNA is undergoing, or it's like later on, it's going to undergo the process of translation of protein synthesis, then it will be forming the beta galactosides, the permease, meaning the enzymes that are all protein. Now remember one thing, students, majority, I think almost 98% of the enzymes that are there, they are proteinaceous in nature. There may be some RNA enzymes, but still other than that, there are no enzymes that are any other nature. Enzymes are mostly proteinaceous. Is it clear? Has everyone
Yes, ma'am.
understood this whole entire concept?
Yes, ma'am.
Okay. So after that, we come on to the Human Genome Project. Now, Human Genome Project, this was a very, very huge project that actually told us about how the human genome is like. Genome means the set of genes that are there in every single human being. How is it different from one person to another person? How is it so huge that it was such a mega, you can say, project and stuff? So that is what is dealt with here. So genetic makeup of an organism, meaning the genotype of the organism, it is basically in the DNA sequence. So the difference in two individuals will naturally be reflected in differences of their nucleotide sequence, meaning how the genetic sequence is present in different person's body. Like see, all of us are one organism, we are one species, but still we're very different from each other, even though the basic genes that we have will be present in all of us. Like, there will be no difference in that. But how do we know that one person is different from another? And how do we know that one person has this set of enzymes being produced in them and the other have another set of enzymes? So that can be known using the Human Genome Project. So we can know how two individuals are differing if we know the entire human genome in general, right? So with the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, meaning, see, genetic engineering is a very huge branch of science. Okay, here you can take a piece of DNA and you can make thousands and thousands of copies, and that is called as cloning. Okay, like how clones are exact copies. So you can make thousands of copies using one piece of DNA. And these techniques, they were very much, you can say, developed and in the market then. So a very ambitious project of sequencing human genome was launched in the year 1990. This year is very important to remember. In which year was it launched? Now, Human Genome Project, it was the international collaborative research program whose goal was the complete mapping and understanding of all the genes of humans. The person who wanted to do this, they wanted to know every single gene and what they are present for, what was their use. All of that had to be done under this huge project. Okay, so all genes together of haploid sets of chromosome, we call that the genome, meaning the number of chromosomes we are diploid. We have two sets. So that one set, because the two sets are there, so it's like exactly the same thing. So that one haploid set of our body, meaning the first 26 chromosomes, sorry, 23 chromosomes, they are our genome. The Human Genome Project, it was called as the Mega Project. It was a 13-year-old project. Imagine, 13 years were taken for sequencing the human genome. It was coordinated by the US Department of Energy and the National Institute of Health. Soon, the Wellcome Trust UK, they joined the project as a major partner. Additional contributions came from Japan, came from France, came from Germany, China, and other countries as well. And this project was ultimately completed in 2003. And why is it called as the mega project? This part is very important. Like they can directly ask you, like, why is Human Genome Project referred to as a mega project? So here are the 5 reasons why. The first one, this whole entire project was extremely expensive. So huge cost estimation or estimated to be at that time was 9 billion US dollars. Imagine the whole entire project upon completion required 9 billion US dollars. So the cost of sequencing per base pair, like one base pair, was costed around US $3. Okay, so the whole entire thing was 9 billion US dollars. This was the first reason because of which this is the mega project. Secondly, very large numbers of base pair, that is 3 into 10 to the power 9. Imagine students, 3 into 10 to the power 9 is this, right? This much. These many base pairs have to be identified and then they have to be sequenced. It requires a large number of scientists, technicians, and supporting staff. Okay, these are the 3 reasons. Then the storage. Now, once you are done with doing the project, then you have to store the data. So the data had to be generated and the storage had to be done. So this required 3,300 books. Each of those 3,000 books containing 1,000 pages and each page having 1,000 typed letters. However, high-speed computational devices for storage, retrieval, analysis, all that made it easier. The 4th point. And then the last, the science of bioinformatics also developed during this period and it helped the Human Genome Project. So because of all of these reasons, the Human Genome Project is called as a mega project. It's grand, it's huge. Any doubts up until now, students? No doubts? Also, one more thing. Just for today's class, I am excusing you because some of you say that you're not well. Some of you say that you have internet issues. This is just the excuse for today's class. I will not take it from the next class onwards. If you people don't have your video switched on, I will not be taking the class. So before the next class, please have your device fixed, have your internet connections checked out, because you people come here for 1.5 hours and that's it. And you don't pay attention, you don't speak in the class. So that is something that I am not going to accept. So today I'm leaving. It's okay. You people have your issues. That's okay. But from tomorrow's class or from the next class, whenever you people have it, if you come here, you switch on your video and you sit. I don't have to speak every time because this is a basic thing. If I don't see you, how will I teach you? Okay. So for today's class, I'm giving you an excuse. It's okay. But from the next class, I will not take it. Okay. You people have to switch on your video. You people have to interact. If you don't interact, this is your class 12th. You are the senior most. So then you people are going to have a lot of problem if you don't study nice because you people have your boards. So take your studies seriously and do as you're asked. So just for today, I'm giving you an excuse, but from next time, from the next class, I will not take any excuse. Okay? Is it clear to everyone? Yes or no?
Yes, sir.
Okay, so furthermore, the goals of the Human Genome Project. Now let us see their goals, what they wanted to do as their project completed. The first thing is that they wanted to do the identification of all 20,000 to 25,000 genes in human DNA. However many genes are there, they wanted to identify those genes. They wanted to find out what is the function of this gene, what is the function of that gene. That is what they wanted to do. Later on, they also wanted to determine the sequence of 3 billion chemical base pairs. Imagine 3 billion chemical base pairs that are there in human DNA. They wanted to find out the sequence, like how people have seen A, T, G, C, all of that. So in which manner is this arranged? whether A is here, G is here, T is here. So whatever is the arrangement, for 3 billion of that, they wanted to know. Then after doing all of this, after getting all of the data that they are supposed to, they wanted to store this information in database. Okay, they wanted to store it so that whoever wants to study it, whoever wants to do some sort of, you can say, research, they'll have this data available. Then they wanted to improve the tools for data analysis. They wanted to find out that if there's any person, even if you are there and you want to study about it, you want to see how many genes are there, how many structures can be formed, what we are made up of, how many base pairs are there. So for data analysis, the tools are available and they wanted to improve that. And then once they are done with, like once they are filled with the knowledge of all of it, they wanted to transfer like the related technologies to other sectors, such as in the case of industries. Like how you people know that there are medicine industries, right? There are industries that make medicines, there are industries that make vaccinations and stuff. So if, let's just say, if there's any company that would know the way the human genome is there, so they can make better medicine so that they will be more effective according to the gene that the human being has. Okay. After that, then comes the LC. LC is like the E stands for ethical, L stands for legal, and the S stands for social, and the I stands for issue. So they wanted to solve ethical, legal, social issues that are related with the human genome. Okay, and how to use it for the benefit. Bioinformatics. It is the close association of human genome project with the rapid development of a new area in biology. So this was also developed accordingly. Now at that time, humans genome was being studied. This whole project, the whole entire human genome project was for humans only, but other organisms, they also were tested. They also were studied. So non-human organisms, meaning all those organisms that are not human, those DNA sequence can lead to an understanding of their natural capabilities and how, again, you people know that we humans, we are so clever that we can use anything for our benefit. So now, once we are done finding out the human's genome, now let us find out the genome of other organisms. For what? So that we can use them for our benefit. Like we can use it in the healthcare, we can use it in agriculture, we can use it in energy production, environmental remediation and stuff. So this line is very important. The non-human model organisms such as bacteria was sequenced, yeast was sequenced, Sinorhabdidis elegans, which is a free-living, non-pathogenic nematode. Okay, that was sequenced. Drosophila, the fruit fly, and then even the plants were also studied, like the rice and Arabidopsis. So these organisms, they have been also sequenced, and these are non-human models. So these are non-human models that are also studied. Okay, and see, this is like the data that they found out. If you people want to write it, you would write it, or if you don't, it's okay. So E. coli, 4.7 million base pairs are there in it, and the total number of genes in E. coli are 4,000. Saccharomyces. Saccharomyces cerevisiae is the scientific name of which organism, students?
Can you repeat?
Saccharomyces cerevisiae is the scientific name of which organism? Hmm? Come on, people don't remember. Yeast is the scientific name of yeast. So yeast has 12 million base pairs present and the number of genes is 6,000. Then comes the Sinorhabdidis, the nematode. It has 97 million base pairs and 18,000 genes present. Drosophila, 180 million base pairs, 13,000 genes present. Arabidopsis, 130 million genes, 25,000 genes present. And then Oryza sativa, for 30 million and has 32,000 to 50,000 number of genes. Is it clear, students?
Yes, ma'am.
Now, see. The methodology. What did they use for this human genome project? So it had two approaches that were involved. The EST, that is expressed sequence tag, and sequence annotation. See, even if you people don't remember what it is, you should know what were the two major approaches. Okay, these names you should. The first, identifying all the genes that are expressed in us in the form of RNA instead of being expressed in the form of DNA. Then sequence annotation is then sequencing the whole set of genome that contained all the coding and non-coding sequences and then assigning the regions with functions. So first what they did, they took the DNA, they cut it into fragments, they cloned the DNA, made multiple copies, and then they sequenced it using the DNA sequencer. This is like the the 4 steps that they follow, the basic 4 steps. Okay. So for sequencing, the total DNA from a cell is isolated and then it is converted into fragments, meaning cut into small fragments. And then they were cloned. So the cloning resulted into amplification. Means clone means making multiple copies of one thing. So the cloning also took place. Okay. And then the commonly used host at that time were bacteria and the yeast. So we call it as bacterial artificial chromosome and yeast artificial chromosome. So these are the, you can say, the chromosomes that were used to create this whole entire process. Okay. After that, the fragments, they were sequenced using automated DNA sequencer. Sequences were already there. It is a machine. And that worked on the principle of the method which was developed by Frederick Sanger. Okay, so he determined the amino acid sequence in the proteins and he arranged them on overlapping regions. Okay, so DNA sequencer was used to sequence the gene. Okay, ultimately, the sequence of chromosome 1 was completed only in the May 2006, which was the last of the 24 human chromosomes. Okay, that contained 22 chromosomes and XY to be sequenced. So the sequence of the chromosome was completed on 2000, like in the May 2006. And then ultimately, the features of the human genome was given. Okay. So up until now, do you people have any doubts, students? Anything you want to ask?
No, ma'am.
Okay. Okay. So what is the last thing you people wrote down in the previous class? Hmm?
Ma'am, last class I was absent, so I don't know.
Okay, whoever was present, please tell me. Marwa, what is the last thing you people wrote down? This part, this operon concept was done.
No, ma'am, I was writing operon concept.
Okay. Okay, now you people can start writing from here. And once you are done, unmute and tell me, ma'am, scroll down. Okay.
Okay, ma'am. Done, ma'am.
Everyone's done till here? All of you are done, students?
Yes, ma'am.
Oh, very good.
Done, ma'am. Ma'am,
Yes.
ma'am, can I leave? I have exam.
Okay.
Thank you.
The rest of you are done till here. Shall I scroll down?
Yes, ma'am.
Yes,
This
ma'am.
diagram you've drawn?
Yes.
Oh, very good.
Ma'am, can you scroll down?
Yes, all of you are done.
Yes, ma'am. Done, ma'am.
Done? Both of you are done, students?
Yes, ma'am.
Oh. See, this chart you don't have to note down. It's not important as far as the questions are concerned. Just note down till here. Like write down. All right, students. Let's just keep it till here only for today's class. Do you